All right, so welcome back here on Thursday. And we're going to kind of pick up right where we left off as far as talking about enzymes. So last time, yep, we got all the way through looking at the basics of what enzymes are, how they function, what they do for chemical reactions, and then talking a little bit about names and the terms we use to refer to the different parts of an enzyme, like the active site and things like that. What we're going to look at today is the different factors or things that affect enzymes, that affect how well they work, about how fast the chemical reactions that they participate in are able to go. And in so doing, we should be able to make um, some pretty solid extensions into the world of things or phenomenon that we have seen or we have experienced biologically speaking, in our own bodies that have to do with how enzymes might have been affected in a positive or in a negative way. Alrighty, so factors that affect enzyme function. These are the ones that I want to make sure that I go through with you. All right. Um, so the enzyme concentration and the substrate concentration can both affect the rate of an enzyme catalyzed reaction, how quickly or how slowly the enzyme is able to completely turn all the substrate into product. Environmental factors like temperature, pH, and salinity can also affect the enzyme's ability to function because they can change the shape of the enzyme. They can interfere with some of the interactions in the secondary and the tertiary structure. And then other substances like activators and inhibitors can directly attach to or interact with the enzyme to affect its ability to function the way it should. And so this picture is kind of a computer rendition of the catalase enzyme, which we're going to look at, or we did look at today in the hydrogen peroxide experiment. Um, and so that's kind of just a graphical interpretation of that. So we're just going to go through these, and we're going to talk about how they affect the ability of the enzyme to work. So normally we would be doing the Enzyme Catalysis Lab, in which I wouldn't tell you how all these different factors affects the enzymes, but we would do an experiment, and you would be able to choose what uh, factor you'd like to test, and then you would end up presenting the data to the rest of the class about your findings. So enzyme concentration. If we plot the reaction rate, how fast the reaction the enzyme is participating on the y-axis, and the concentration on the x-axis, you can see this this almost logistical curve, this logistic curve like this. All right. So it seems that it is kind of exponentially increasing, the reaction rate that is, and then as the concentration gets higher, it kind of starts to plateau. And so we're going to talk about that. So in general, as you increase the enzyme, you increase the reaction rate. The idea is that the more enzyme there is, the more frequently the substrate and the enzyme collide, and the substrate is turned into the product. I think that seems pretty straightforward. But the idea is eventually it levels off. If you saturate the reaction with enzyme, the substrate becomes the limiting factor. And not all of the enzymes can find substrates. And so the enzyme and the substrate are coming together and reacting as quickly as possible. And adding more enzyme really isn't helping because the substrate is what is in limited supply. So in general, increase the enzyme concentration, increase the reaction rate, but up to a point, and then we'll have kind of that diminishing returns model. We're going to see actually much the same pattern if we change the substrate concentration. So if you look at this graph here, it looks almost the same as the one for changing the enzyme concentration. So again, as we increase the substrate concentration, the reaction rate is going to stay the same. More substrate, just like enzyme, more frequently we're going to have substrate and enzyme come together to form our enzyme substrate complex, and we'll have a reaction. But the reaction rate again levels off because after some concentration, after the concentration of substrate raises past a certain point, all the enzymes are engaged with substrate all the time. The enzyme is saturated. All right, we say it's saturated. Um, the enzyme is working as fast as it can, and they are in limited supply. So we end up with a maximum rate, this plateau up here. The next things we're going to talk about is environmental effects. So let's think. Substrate and enzyme concentration both behave the same way. We increase the concentration, we increase the rate of the reaction to a certain point when the reaction becomes saturated, and either concentration of substrate or concentration of enzyme becomes limiting. Here is the graph of reaction rate and temperature. Now, one disclaimer is that this is just a random temperature that I picked. It's not to say that all enzymes have an optimal, and that's what we call it, an optimal temperature at 37 degrees. So, every enzyme has an optimal temperature, and that's what this green line represents the peak of this reaction rate. It's when the greatest number of molecular collisions occur. In the human body, most enzymes have that optimal temperature between 35 and 40 degrees centigrade, keeping in mind that about 37 is 98.6 body temperature. If you increase heat beyond the optimal temperature, because you're like, well, we remember from like the molecular theory that if you're increasing the temperature, you're allowing those molecules to move faster. And if they'd move faster, wouldn't there be more collisions? Yes, but increasing the temperature past the optimal temperature will start to, and here's that word again from biochemistry, start to denature the enzymes. Though that heat will start to disrupt the tertiary structure, the secondary structure, interrupt the hydrogen and ionic bonds between our groups, and then the enzyme is going to lose its tertiary shape. Its shape is going to change, and the substrate won't be able to fit into the active site anymore. Yep, it won't be able to fit into the active site anymore. If we make the enzyme too cold or colder than the optimal temperature, the molecules will start to move more slowly, and there will be a decrease in collisions between the active site and the substrate molecules, and so the reaction won't be able to go very quickly. So kind of a bell-shaped curve to the optimal temperature for enzymes, and different enzymes will have a different optimal temperature depending on the environment where they're specialized to function. So, for example, there are enzymes that thrive and live in hot springs. This isn't actually a hot spring. This is a geyser in Yellowstone, but same type of environment. under the ground as a hot spring. Their optimal temperature is 70 degrees centigrade, so almost boiling, whereas humans, most enzymes are around 37. Their optimal temperature, all right, has matched the environment where they live. So those enzymes in those organisms have evolved, they are there so that they work best in the environment that the organisms are found in. One of the questions that a lot of people might ask, though, is how do cold-blooded animals do it? Warm-blooded animals like ourselves, endotherms, are able to keep their body temperature constant. And so those um, enzymes can stay at their optimal temperature since our bodies stay pretty much at the same temperature all the time. But how do things like snakes do it when their body temperatures change depending on the outside temperature or the ambient temperature? Wouldn't their enzymes, when it's cold, be uh, going way, way, way too slow to support life? And wouldn't they all get denatured if they're out somewhere when it's hot? How do you think this happens? I'm actually going to talk about this in our next Zoom meeting on Friday. So if I forget, remember to mention this because it's something I want you to think about. All right? It is a critical thinking type question. All right, so just like temperature, pH or acidity and alkalinity can affect how an enzyme works. And if you look at this graph, it's much the same case as temperature. There are optimal pHs, and too far one way or the other, too acidic or too basic from that optimal pH, those enzymes will begin to be denatured. Their ionic and hydrogen bonds will start to be um, broken, interfered with by the pH of the solution, and the enzyme will cease to function as well. So look at the different, these are different enzymes inside the human body and actually in the digestive tract. So pepsin in the stomach has an optimal pH below 3 because our stomach, with its hydrochloric acid and everything, will have like a standing pH of below 3. Whereas trypsin, which is found in the duodenum of the small intestine, has an optimal pH of a little over 8 because a very basic substance, sodium bicarbonate, is dispensed into the duodenum by the pancreas to kind of neutralize the stomach acid after there is the gastric emptying. Oh, wow. So trypsin, an enzyme that breaks down the food in the duodenum, has an optimal pH in a more basic environment. Even though these two, trypsin and pepsin, are found very close to each other geographically in the body. So again, pH changes in the changes in pH can affect enzyme function by adding or removing hydrogens because a basic solution or basic environment will remove hydrogens and an acidic will add. And it disrupts the bonds in the 3D shape in the secondary and the tertiary structure of our enzyme proteins. And so it'll denature them, we'll have a shape change, and then substrates can't fit in the active site as well anymore. Most human enzymes will have an optimal temperature between 6 and 8, but it depends on the conditions. So like I said, pepsin is found in the stomach, and its optimal pH is in between 2 and 3 because that is the acidic conditions in the stomach. And then lastly, salinity or the salt concentration is the last environmental factor I want to talk about. And you'll see much the same graph for much the same reasons as acidity and temperature. So changing in salt concentration can affect the ionic bonds within a protein. The salt breaks into positive sodium ions and negative chlorine ions, and they will be attracted to the different positive and negative um, parts of the ionic bonds in our tertiary structure of a protein and cause, again, denaturing, which will decrease the enzyme's function. All right, if you think about it, enzymes are intolerant of extreme salinity. So just like the Dead Sea, that's why nothing grows there. It's called dead for a reason. All right. And lastly, I want to talk about some compounds or substances that both help enzymes function and inhibit enzymes function. So there are some things like called activators that help an enzyme's function. Certain inorganic, meaning doesn't contain carbon, inorganic substances or activators we call cofactors. So cofactors are inorganic. They're non-protein, inorganic compounds and ions like magnesium, potassium, and calcium, and zinc, and iron, and copper. And this, if any of these sound familiar, this is what a lot of people call, all right, minerals, all right, or you might take, or vitamins. You might have these in your multivitamin. Most of the reason that those are important for you to take or to have enough of in your diet is because they facilitate proper enzyme action in your metabolic processes. So, for example, iron is really important in hemoglobin in your blood cells for it to work. So these cofactors facilitate or make enzymes work better, and so they're necessary for your body to take in and for you to have in small amounts in your diet. Other activators that are similar to cofactors, they do similar things, but their organic molecules are called coenzymes. They bind temporarily or permanently to the enzyme near the active site, and they help out, again, with the enzyme catalysis. So many vitamins like B3 and B2 and coenzyme A um, are all coenzymes. They help enzymes in your body work better. So those are called activators. And then there are inhibitors, which, like their name suggests, inhibits or reduces the enzyme activity. And there's kind of four different groups of them that we'll talk briefly about. So competitive inhibitors compete, as their name would suggest, for the active site. They can fit in the active site just like the substrate, but they aren't catalyzed. They're not broken down, and so they sit there and block the enzyme from being able to bind substrate. So, for example, penicillin blocks enzymes in bacteria that bacteria use to build their cell walls. And that's how they eventually destroy bacteria. They prevent them from being able to renew their cell walls, and they eventually disintegrate and die. Um, a competitive inhibitor that is used to treat alcoholism blocks enzymes that break down alcohol. So, um, it when somebody drinks alcohol, it causes a severe and negative reaction in their body, so that they won't drink it any get more. Um, and competitive inhibitors can actually be overcome by increasing the substrate concentration. All right, if you saturate a solution with substrate, they're just always able to outcompete the competitive inhibitor for the active site. There's just way more substrate than competitive inhibitor, and so they kind of win out. Non-competitive inhibitors inhibit enzymes, but they bind to a site other than the active site. It's called the allosteric site. And binding to that site changes the shape of the active site. There's a conformational change so that the enzyme can no longer bind with the substrate, right? So some anti-cancer drugs inhibit enzymes that are involved in DNA synthesis by being non-competitive inhibitors. Um, Cyanide poisoning also does the same thing. So it irreversibly binds to a enzyme that's necessary in cellular respiration, and it isn't able to catalyze its substrate to products, and so ATP in your cells isn't able to be made anymore. There are some competitive and non-competitive or allosteric inhibitors that we call irreversible, which means they permanently bind either in competitive inhibitors case to the active site or in a non-competitive inhibitor to the allosteric site, and there's a permanent shutdown of that enzyme, right? Permanent conformational change. There's no way that enzyme can ever overcome that inhibitor. It's just no useless anymore. So many nerve gases and, for example, insecticides work this way. Okay, it it binds irreversibly to the allosteric or the active site, and they aren't able to be removed. They're permanently stuck there. Let's see. I think that's where we're going to stop today. Just looking at the rest, that's where we're going to stop. I think we've gone far enough, and the other stuff we'll kind of finish up tomorrow when we look at ATP.